And Karina is the PI on this, one of the PIs on this project, on the immunology project. But she was sorry not to be here. She had some surgery that had to be moved up, so she's going to be fine. But she's sorry she can't be here. The other thing is, we're going to give you a little bit of background about the database itself, what we do, why we do it at Los Alamos, and which has a rather different mission, broadly speaking. But obviously, like this project, there are a lot of people involved, and we're grateful, of course, to all those people, and in particular to NIH, who without whom this work would not be possible. Is there a pointer?
This one should.
That's a small point.
Let me see if I can try and point it out.
Okay. Excellent. Okay. So first off, just the beginning thing. Today I'm going to be talking about the HIV. The HIV immunology database. Tomorrow we'll be talking about the sequence database. And can you hear me okay? Am I close enough to the mic? Okay, good. And we'll talk about, broadly speaking, what we do, which is interestingly similar and different to what you've been hearing about so far. What's done here. We'll talk about several of the tools and our way of doing database access. One key point is we have a bunch of tools for processing immunological data and sequences, some of which are specific to HIV, some of which are really absolutely general, and some of which are mostly for HIV, but can be applied with caveats to other pathogens. So today, we're going to hope to get to at least through catnap before the break, and then after the break, we'll be talking about some of these additional tools and some cool stuff and brag alert, a little bit of a nice story on vaccine design. So the history. So Los Alamos, for those of you who aren't familiar with it, is a nuclear weapons laboratory. It's the birthplace of the atomic bomb. It built out in the godforsaken wilds of northern New Mexico because Robert Oppenheimer went there as a boy, to a boys' camp school / as a kid, and there was nothing there. What a place for a secret project. And now it brings in $2.5 billion a year, most of it to northern New Mexico. Huge deal, 12,000 people, mostly working on defense. However, however, in the 50s, the Department of Energy, for whom the lab was run, started worrying about radiation effects on genetics, and therefore started worrying about genetics. And they had big computers there, and they had people who started thinking about ways to use computers to deal with DNA sequence data, and that became available. And that led to the establishment of GenBank. By the way, GenBank started in the same group where we are now. And a spin-off of GenBank was the HIV sequence database and immunology database. So brag sheet again, we get a lot of hits from the research community. We get a fair number of citations, and we've got a resource which is important for the HIV community. Now, is there anybody here who cares about HIV at all? Anybody? Give me a hand. Okay, 1, 2, 3. You don't count those. Okay, 3, got 4. So that helps to focus things, because there's a certain level of basic knowledge that we can assume when I'm giving this talk at an HIV meeting, but different here. But the point is, again, that we have a bunch of tools, and many of them are general purpose. So, I have a feeling this is okay. So if you go here, hiv.lanl.gov, Los Alamos National Laboratory.gov, You'll see this page. This is, we'll go back back and forth to this at various points during the talk. Key point is the two databases we talked about today are the sequence database and the immunology database. These guys are also present but less active at this point. And whenever you come here, you will say, oh, what's new? Well, this is what was new when we made this slide 3 years ago. There's been new stuff since. So again, the sequence database was founded in 1986 by Jerry Myers, who noticed when the first HIV envelope sequence was generated. That was interesting. Looked at it. Second one, he saw that, oh, that's considerably different. And the third one was substantially different from those two. His basic take was, we may have a problem here, and started collecting additional sequences. And again, Betty Corbett, about whom I can't say enough that's nice. If you get a chance to meet her, you should. She's a spectacular person, and the person behind it, still active in vaccine design and databases. Found the immunology database as an integrated part of the sequence database. And the integration is really key because unlike other pathogens, or to a greater degree than other pathogens, the process of HIV infection is both in the individual patient and epidemiologically is a very deep interplay between immunology and virology, because the ecosystem of the virus is the virus is essentially adapted for long-term persistence in the context of an immune response. So there's a very tight degree of integration of the immunology and the virology. And this has led to the development of a bunch of tools to reflect that integration. I'm going to spend a lot of time today, the biggest portion of the talk here, on this tool called CatNap, which is essentially a way of trying to combine the data we have from large-scale experiments based on neutralization panels of different viruses, different variable viruses, and different antibodies that we know about. Great tool. Karina developed it primarily. We're also going to talk about diversity and variability of epitopes, because for our again, standpoint of HIV, variability is the thing, and so we need ways to address that. And I'll talk briefly, genome browser, which is just a nice way of integrating features on the genome, everything we know about the HIV genome and the immune response to it, with a view of positions of these features on the genome. And a variety of, and Elizabeth will talk about a variety of other tools and the search tools. Point again, many of these tools are broadly applicable. If you go to the tools page from that first page, you'll see this list. And this is just a sampling. This is most of the immunology-specific tools, some of which are colored in red because really they don't make sense outside of HIV. Some of them are most useful for HIV, but can be used elsewhere, and some are just plain general. So the story I'm going to talk about later in the day is an example of the tools that we developed for HIV that are useful elsewhere, and that's the Mosaic Vaccine project. And so to date, these different vaccines have been made with this method. All of these have shown some degree of efficacy in trials, and right now I'll talk about the HIV application of this tool, which currently is in phase 2B trials in Western Africa. That's the one that's most advanced. We also have a series of other virus databases, the hepatitis C database, and a hemorrhagic fever database, which contains 80 different viruses, as well as a filovirus sequence database, which, surprisingly enough, in 2014 there was an additional demand for filovirus data. There was just some. It happens to be people began to pay attention, a little more attention to this, and that gave us the opportunity to upgrade this data a little bit. But the key point, it's a database, so we have to get data in, and Elizabeth Fung, who's up in the corner and coming down shortly, will be talking about how the data actually is put in our process, which is not dissimilar to what the folks here do. Oh, that's not right. Oh, well, that's interesting. I'm sorry, I just broke it. I want to get the presenter view up and instead, so. Yeah, I want to do a presentation. No, this is on.
You can just close that one.
I think it
On the desk.
You can just close that one as well, so it doesn't have to get. You're right there, very bottom. Yeah, double.
That was very strange. My apologies. So, okay. So, at this point, I'm going to turn it over to Elizabeth, who will walk you through, and you can go follow along live, walk you through basically what's here and how it gets there. And I should say, by the way, feel free to jump in at any time, particularly now that Elizabeth's talking, it's not me, because she handles that. And I may be jumping in at various points too. So, and I would encourage you to go ahead and follow along with the links and go ahead and explore what you see, and shout out if you have questions.
I am Elizabeth Sharon Fung, and I am one of the annotators on the immunology side of the database. And I know I said I'm an annotator, but a few days ago, my child came back from school, an 8-year-old, and said, "What do you do at work? Are you an agitator?" So, don't give me grief. Okay, no, but ask questions, please. So I had a couple things to say. Karina sends her apologies, just as Will said she was planning to be there the day before we left, and doctor said no. So she's not here, but this is her work mostly, a lot of stuff, and so if you have questions either of us can't answer, please give us those questions, and in all probability she will have the answer, and we can definitely get back to you on that. The other thing is, as we go through, please let us know if there are any suggestions you have, because a lot of good things come out of users' suggestions, and a lot of the tools and things have gotten better because of people's suggestions to us or questions. So feel free to jump in and ask if you have any questions. So going back to the slides in the presentation here, this set of databases has an immunology and then a sequence database, and they're very well linked. You can get to almost anything from the immunology database, and if you can't go directly, you can get to it by linking through to the sequence database, and the converse is true too. And so they're linked, they inform each other, and of course, as an immunologist, you want to know what's happening on the sequence end of things too, and so it's really good for planning experiments if you're an experimentalist or not. So these are really well integrated. That's one of the strong points of the database. The other thing is in the immunology database, we have notes. And these notes have been generated by manually going through the papers. This is not just the abstract of the paper. It is actually not the abstract, but these are notes that are relevant immunologically. And so we'll get to that a little later. And then I'm just going to head on to the next slide because I think this is the main page, the mother page of the hiv.lano.gov. And here's a little note for you. You will see the URL that, whatever tool or search interface I'm talking about, the URL will be on the slide. And if you want to follow along, just copy paste it into your browser and look. Now in general, I'm supposed to say that the URLs are all in pink, but I'm saying that at a slide where it's in blue. And there's one other slide where it didn't get changed to pink, but otherwise it should be right there. So if you look at this, you'll see there's these buttons right here. And there's the vaccine database, which is not being actively updated. It's still there, but it's not being maintained and updated. The other viruses database contains mostly hemorrhagic fever viruses and then Hep C, HCV. And while it's not, the immunological part of it is not maintained, every month new sequences are uploaded, but otherwise it's not being maintained. But you can find the current repository of sequences for the hemorrhagic fever viruses. Also, as Will mentioned, in 2014-15, when due to the Ebola outbreak, there was renewed interest, and so there are some very good current alignments for Ebola in particular up on this other viruses database. But what we'll be talking about today is the immunology database, and we will also talk about the sequence database as far as some tools and other things, but most of it will be covered by Will tomorrow. Okay. So let's go on, and this is what you would see if you clicked on the immunology database button. This is the page that would come up, and just as the IEDB database folks said, and that actually reminds me another thing I forgot to say. Thank you IEDB for inviting us, and also thank you NIH for funding us. But so up here you'll see that there's a drop-down menu, and from there you can essentially link to anything you want, tools, resources, sequences, whatever you want. You can go back to sequence database by pulling this down, and you can go back to the sequence database. Remember, we're in immunology right now. And also, everything also comes up as a list here, okay? So in the arrows in red, you can see that there are multiple ways to the database products and tools. And this is just like IEDB. You might find one certain method to find something, and then you find the same data again elsewhere, and this is because they're linked. So if you were to do an antibody search, you would click here, or you would go from the drop-down menu on search here. And if you were looking at database products, you could go to a table for epitope variants, or a table for epitopes, or neutralizing antibody resources. And remember, these are all HIV, okay, over here. However, if you went to tools, you would find a lot of tools that were general use. And some of these tools are really nifty. Output is a wonderful figure. You can color code it. And so it's actually useful for non-HIV people too, just to produce information or figures that you might want. Down at the bottom, you'll see here we do have some other links. There are some SIV epitopes, and there's also some large bits of data that are relevant to HIV studies that were so large and so good that we just asked the authors to give us basically their tables, and we've made them look good and put them up there. So you can actually go straight to the data there on the table, and it's usually downloadable. Okay. Another thing that we can look at, and this is from the same kind of page, is you could look at, if you were looking down here, you could look at different summaries of the data. So you could look at epitopes as maps, or you could look at them as tables. You could also, as I said earlier, look at other tables from other groups, okay, with large-scale studies. So let's go on here and let's just look at what it would be like to do, say, an epitope Ctl search. And by the way, the Ctl searches, whatever I tell you, is exactly the same for helper T cells, epitope searches. So it's not a different, it's different data, but same way of getting there. Okay? Okay, here we go. This is what, you know what, let me go back and reorient you. So if we were to search for Ctl epitopes maps, this is the kind of thing we would get. And this is the way we denote them on the maps. And the maps are interactive. This is just a very small slice of the HIV map, and this is actually, here it shows you all the different epitopes that map to this particular sequence of HIV. Now what we do on the database is we use the same reference sequence, it's called the HXV2, and what that does is to help clear up numbering systems so that everything's aligns to the same place and everybody knows what they're talking about when they come up with a number or a certain set of coordinates on the sequence. Only epitopes that are 14 amino acids or less are shown on this map. Anything larger than that is not shown on this map. And you can see that each epitope is, do it up higher, I'm sure it's so lower is good, but each epitope here is bounded by these two arrows and the line. And it's also referenced by the HLA that restricts it. So that if you have two that are the same location and same size, but they're two different HLA that restrict it, then it's given twice. However, if there is a minor difference in sequence, say over there, then it's not shown twice if it's the same HLA. But because the map is interactive, you can just click on it, and then you'll get to another page where it shows you all the different sequence variations, because HIV is so finicky and varying, and it's just intensely crazy. And because of that, there will always, pretty much always, be a variant, and we have that curated as well. So these maps are interactive, and it helps us to get them, to get to the individual sequences. This is another way that we have of showing the different epitopes that have been curated and annotated into the database, and it's the tabular format. For Ctl epitopes, there are two tables. One table is just a list of every single epitope that we have annotated that is a Ctl epitope for HIV into the database. And so you'd see them as these lists saying which protein it's from, what the location is from, blah, 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 subtype and everything else. And you can see that some of them have, like here, they're variants of each other, okay? However, there is another database and another table, I'm sorry, and that one is called the A list. And that one is very important for experimentalists because it has the epitopes that are A1 class, well experimentally documented, and you can be sure that they follow different criteria that Christian Brander and colleagues who curate this particular part of it have set up. So it's gold standard epitopes that have been proven to be epitopes, and it's a good summary for those who are planning experiments and other things. Okay. We also have variants on these epitopes in different tables, and those can be got to by clicking on the variants list, which I showed you on the front page of the immunology tables. And then there are, like I said, other studies which have polymorphisms, and you can get to by clicking on the bottom links of the page. I'm not going to go back there because I know most of them are HIV people. So how would you search for an epitope if you were looking at it? There are different ways. This is what the search page looks like, and this is a Ctl search page. Those T helper cells, T cytotoxic cells, but the T helper cell page is exactly the same, so you would be able to search it the same way. So what you could do is you could put in, say, favorite epitope to search for. Depending on how many entries there are, you would get different hits on it. For example, if there's a very common epitope in HIV called SL9, and if you put that in, you'd have 285 entries. So you would really want to narrow that search down. And we would suggest that you do that, and you could do that by, say, adding a keyword called escape. these keywords are given down here, and something to note about these keywords is that these are not just the keywords that are published with the paper. These are the keywords that we have determined from a careful reading of the paper. So pretty much if you're searching for something, you can find it by looking up keywords and then your epitope for sure. There are also many other parameters that you can search on, and all of these together will give you a really good, well, you can see in the next slide from the result of the search, but it gives you really good context for that epitope immunologically as well as epidemiologically. So that's a really useful way of doing things. You could also search really narrowly, for example, and we have the search down here. This is the epitope IW9, and if you couple that search with, say, author, and the author's name is Pille, and if you put those two together, then you get one hit. Is that right? It says found one matching record. So you get one hit, and you have this, and you can look at the results. This is the way the results are given. This is all the data that can be categorized easily. And then we have the notes. The way the notes are structured are, the first note is a very short, succinct summary of the paper, the whole paper, and the point of the paper. And then the second note is usually about that epitope of interest. And then we do have future notes because sometimes there are other important parts within that paper that we want to talk about. And if you go down to the bottom, there's a PubMed ID. And if you're interested in seeing what other epitopes were curated from that particular paper, you can go straight to it from here too. Okay? Now all these over here are interactive links, and they give you sort of definitions on what does the keyword mean, what are the different experimental methods and things like that. But if you look on the right side, for this particular paper, there are several links that you can go to. You can go to an epitope map, you can go to an alignment, and you can also go to variants if they exist for this, and there are a lot of variants. I already showed you what the maps look like, and we can go ahead and see what alignments would look like. And this is, if you click the alignment button, this is what you would see. And this comes out of a result of a different tool that we will touch on called Quick Align. You can see here that that particular epitope maps to GAG, which is a protein of HIV. And then you can look down here and you can see that it gives you the location on the HIV genome. And then down here there's alignments. Now remember that there's like tons of sequences in the sequence database. And so when you're looking at your epitope alignment, it aligns them to all the instances of that epitope that it finds. And it tells you, if it's in red, that it's completely identical. But if it's not, it gives you the variation. And of course, there's the name of the sequence. And you can actually download this information as a FASTA or a cluster file, or whatever you want. Not whatever you want, whatever you have. And here we go back. I'm going back to that page, which is the result of the search. And I just want you to look at this again and see that we can look at variants. So let's go ahead and see what those variants might look like. If you click on variants, it will show you a listing of all the variants. In this case, there are just two. And you can see where they varied, and that's given in lowercase here. And you can also see what kind of a variant it was. Now that is a classification, and we've given examples here that we've generated. So was it an escape? Was it a drug resistance mutation? Was it just a susceptible form? It's a variant, but it's still susceptible. Is it just inferred and not really experimentally proved? And so we check these boxes, and you can tell what we say. And however, this is what the author says. In the notes, we'll say if we disagreed with the author, because sometimes it's not exactly what the same, our take on it and what they said. And so you can look at that, and then of course you can always go to the link and look at it in the paper notes. Okay?
I'll just jump in here and say the reason we care about escape is because that's the major mode of viral evolution within a patient, is escape from the immune response. And so again, long-term persistence of the virus, you can see Ctl epitopes coming on, CTL response become active, and then the virus evolves in response and generates escape. And this can happen really very quickly within a matter of 2 or 3 weeks from infection to get complete population replacement. Likewise, in the long term, epidemiologically, we've seen adaptation of HIV, of circulating HIV to the predominant HLA types in a particular region.
So this mutation-based,
I'm sorry, you have to be louder.
you're saying like mutation pose like pressure on virus to evolve, right? These mutation is this virus mutation.
Yeah, the mutations are
They become mutant because the immunological pressure from host, right?
I'm sorry, I'm not quite catching you.
So
So the mutations are occurring all the time and they're being selected.
to viral epitope, they get mutated, as you were saying, right?
Yes.
So it is because of the host pressure, immunological pressure by host to get.
Well, the mutations are happening continuously because the virus is error prone in its replication. But the host selection will strongly affect the fitness of typically of the wild, of the initial form, initially infected form. There's a frequency dependent selection. So a common form that is targeted will be disfavored, and mutant forms will then replicate up to the high frequency.
So this information get gathered using a reference system and looking over time how it get mutated.
You can do it both based on a reference genome, but better is to use an autologous sequence from an early time point from that same person. So a lot of people will take the earliest possible sequences and make a consensus of that, and to treat that as a transmitted founder virus. There are complications there, but we'll talk about that separately.
And later on today, that we do have a slide that shows that exactly what you said, a longitudinal study over 3 years, and you can see how different the epitope residues are, the amino acids, and how it changes, and how the relative frequencies of certain amino acids changes. And it's because, you're right, because of the pressure that the virus faces, and then the form that escapes is the form that replicates and outgrows and takes over. So yes, it can be seen, and you can actually follow it, and it is followed in the same patient. And we also have a patient database as part of this, and you can find that patient's sequences or sequences from that patient and check them out if you don't have your own sequences. All right, well, the next thing I would like to go to is how to search for antibodies. And it's kind of similar, but not the same, because of course they're antibodies. And so one of the examples for something you might, what might you do if you wanted to do an antibody search? You could search for a certain antibody, and then you could actually do more with it. And we'll go over this. And that is, you could go through a tool or a resource that we have called CatNap, where you can actually find neutralizing antibody data, everything that's been published about that HIV antibody, and or anti-HIV antibody. And then you could use that, and then you could go on to a different resource, for example, CombiNabber, where you can look up multiple antibody cocktails, including the one that is your interest, and see what their effectiveness is based on neutralization values that you already have from CatNap. And so what I will do is just help you through with this bit of it, with how to search for a particular antibody record. And before I start talking about that, I just want to maybe I'll go back here. I wanted to mention that the HIV database here, this part of the immunology database lists whatever antibodies are in the HIV literature. Some of these, for some of these, we do not yet know the epitope that it binds. It hasn't yet been delineated fully. But if the epitope is known, then it's in there, and you can get to it very easily. Okay? So here's how you would do an antibody search. You can see that there's many ways of searching on them by subtype that it binds, or by different sections, regions that it would bind on the HIV, and you could just search by name too. If you've forgotten the name of your antibody of interest, or you're a little confused, you can list them by type or name and then choose which one you want to right there. Then you get the search. Now right here, I'd like to stop and say that the way that we annotate our epitope databases, the T-cell epitope databases, the Ctl and helper, and the way we annotate the antibody are a little different. What we do for Ctl and helper is every single epitope that is represented in a different publication gets its own record and entry, but not so for antibody. The same antibody can have just one entry, and every note aligned with or set with that particular entry is actually from a different paper. So you could have, for example, you could search for 2F5, which is an old HIV antibody. And if you searched for that, you would get 815 notes, because there's that many papers who have discussed it and that have been annotated in here. However, if you were searching for, say, SL9, you would get 200, which is a T-cell epitope, not an antibody thing. If you searched for that, you get 286 entries. So there are 286 individual entries because there were 286 publications with SL9. But here, if you searched for 2F5, you would get one entry on record with 815 notes, which each note corresponds to a different study. Okay? So there's a difference in the way that shows up, but what it really translates to for a user is, how do I search? So when you're searching for an antibody, if it's a very well-studied antibody, you'll know that already, then it is best to sort of, what's the word for it? To
Narrow down?
narrow your search, thank you.
Drill down, if you like.
To really narrow it down your search by using keywords, and that really helps. Did you have a question?
Yeah, so when we find like one record having 800 references and another one is having just one, so does it lead to the biasness in the literature, like the people are more focusing on that record?
Yeah, so definitely it's a literature bias. That's the only thing. It's just we'll annotate whatever they publish, but it's a literature bias, and sometimes it's because they didn't know about a certain antibody, or like nowadays there's people are talking about bispecific and trispecific antibodies, and you know that wasn't talked about 5 years ago. So, or maybe it was, but not in HIV.
Well, the other thing, the major difference that Elizabeth was talking about between our approach to antibodies and our approach to T-cell epitopes are simply that an antibody is its own thing. There are monoclonal antibodies, but there aren't monoclonal T-cell responses. So a number of different labs will be working on the same antibody. And it is the same because it's identical protein level, but typically the T cells are not so replicable.
Thank you.
So this would be the result of a search on the antibody database, and the example we've used here is 10E8, and you can see that actually 10E8 should come up with one response, but one record, but here you can see 30, and that's because it's so well studied. People have actually made like chimeric antibodies with it, and so everything with the word MP with 10E8 in it comes up. But there's essentially one just plain old 10E8 result, and this is what it would look like. You can see once again that there's many things in it. There's no notes, and there's different, like I said, different papers. Basically, of course, we don't have all 815 here, just two. Okay, and so, but some of the things that you can look at over here is you can link to CatNap, which we'll share with you later, very soon, and then you can link to something called Antibody Features. And these are very useful features that we have. The link to the antibody database features will actually take you to contact residues that are important in the epitope and the antibody site, the paratope. And so there's just information on that from the papers that have been annotated. And then you can also link to patient donor details. So there are some patients who have been extensively studied, several antibodies studied within them, and also, and I didn't mention this with the T-cell epitopes, but we also have donor or patient database linkage there. Because if you're trying to follow a certain patient, you know their code, you can put it in, and you can pull out whatever's been studied about them, whatever has been published about them. On the right side, again, you can link to the epitope map, or you can link to an epitope alignment. And this is because in this antibody, the epitope is known. Like I said earlier, there's a lot of HIV monoclonal antibodies for which it is not known, or even neutralizing antibodies that it's not yet been determined. Okay, let me go back here. If we were to look at what antibody features database links to, this is what it would look like. And so what it would look like is you would get this. Now, you can see this is actually a search because these tools are available in different ways. You can get to it from the antibody search like I showed you, but you can also just simply go to tools and features and look for antibody feature database and then pick your antibody of interest and get to its features. And then it would, if you did that, it would take you to a page that would show the different references, like the different publications, and then you pick the one that you want. So for example, this is Swung 2018, 2012, and under this paper, this is the information that we have annotated about antibody contact residues and why that particular contact residue is important. We also have some information here that tells us about the variability of that position, so like the N, in the epitope. So now I'll hand over to Will, who's going to take us through to other neutralizing antibody resources.
Thanks. So, again, we all still awake? Everybody's awake? Well, those people who are asleep, please say nothing. All right. So you could get to this page going from the front page of the database to immunology, and then to under products, I believe. And we have several things we're talking about. You know, I've been taunting you with catnap. Sorry, I was asked to stay close to the mic. So we've been taunting you with catnap all afternoon, but we'll come to that. And Elizabeth mentioned CommonAber and genome browsers. These are various resources. I'd like to point out that there are two new tools which are going to be, I think, really useful coming up shortly. One is a genetic signature tool, which will take a set of sequences and susceptibilities and do phylogenetic corrections, giving you the signatures of, that is to say, residues that are important for that antibody activity. And another is the filtered forests tool, which is associated with it. This is a machine learning, and people talk about machine learning as though it's terrible, and that's mostly because it is. Just saying. But there are some things it does well, and in this application, it can do a pretty reasonable job of taking a set of a sequence and inferring whether it's going to be neutralized or not. And these the paper is almost